So this is another episode of the Classics in Immunology Journal Club. So you can find that on my YouTube channel. And so if you like this particular series, go ahead and look on it on the playlists of the YouTube site. You should subscribe to it because then the YouTube people are happy. And also click on the little bell right next to the subscription site because then you'll get notifications whenever I do another video. And then also, if you scroll down, you can see all the links to my website and so forth, where you can find all the papers for the classics of immunology from the last 200 years up until pretty recent times. And so you can have a whole library of the classics from my website. So today we're going to talk about a paper from 1968 by a Swiss group. Teddy Brunner, a fellow named Jean-Manuel, Jean-Charles CEROTTINI, and Bernard CHAPUIS. And this paper was published in Immunology, which is a British journal. And the title is, it's quite a long one. I have to read it to you. It's Quantitative Assay of the Lytic Action of Immune Lymphoid Cells on chromium-51 labeled allogeneic target cells in vitro, inhibition by isoantibody and drugs. So this is another methods paper, and this is a classic methods paper in cellular immunology, really describing what became the definition of a cell-mediated immune response in vitro. So to give you a little bit of background on this, Teddy Brunner was he was a very much of an outdoorsman. He was the head of the Swiss Cancer Institute in Lausanne, Switzerland. MAUEL was a student at the time, and Jean-Charles CEROTTINI, the third author, was a postdoc, and he was in the lab next door to Teddy Brunner's lab, and he became interested in what Brunner was doing. And it turns out that what Brunner was trying to work on was as he lays out, or as they lay out in the intro, is that it had been demonstrated that lymphoid cells, when they were sensitized or immunized in vivo, so if you took a mouse, you could immunize one strain of mouse with lymphocytes from another strain of mouse, and then you could take those immunized spleen cells or lymph node cells or whatever, mix them together with the immunizing strain of cells. A lot of times they use tumor cells, and it looked as though that the immunized lymphoid cells were killing the stimulating cells. Problem was that it was difficult to really be sure that what was going on, that maybe antibody and complement might have been involved, and whether or not it was really a real phenomenon or not. And as you know, going back more than 10 years, 15 years to Peter Medawar and his 1953 paper of the graft rejection, this is the kind of thing these people were interested in. They were interested in allografts rejection, and they were also interested in tumor cell rejection. So they needed a better way to really demonstrate this and to quantify it. And it had been originally they looked down the cells at under the microscope and could see some of them dying and some of them living and so forth. And then Brunner had been working on an assay for cloning the target cells. And so for this, he used tumor cells because tumor cells would grow in test tube or in the flask. You didn't need to stimulate them with anything. They just grew all by themselves. And you could then, if you plated them out in a petri dish with soft agar, at a very low density of cells, you'd get little colonies form. And so that was a cloning colony forming assay. And so you could count them. This was good. And others at the time, and this is back in the early to mid 60s, we're talking about now, others had demonstrated that if you radiolabeled the target cells, and that could be done by putting in precursors of DNA like C14, thymidine, and other kinds of things like that they would become incorporated into the target cell. You could wash off the excess radioisotope radiolabel, and then you could look for the release of that radioactivity into the supernatant if the cell got killed. Just a few years prior to this 68 paper, there was a report from Stockholm from Jan Klein's group and Peter Perlman's group from the university, and Klein was at the Karolinska, where you could label the target cells with chromium-51 and sodium dichromate. And they showed that what you could do is you incubate the target cells with the chromium for a while, not just 30, 40 minutes. And then you could wash the cells and the cells would take up the chromium, and the chromium would somehow attach itself to macromolecules inside the target cell. So you could wash off the excess, and now you had a radiolabeled target cell population. And that's what these people, Brunner and his team, really solidified, and that's the subject of this particular paper. So he goes on to the materials and methods and how he went about doing this. And this is important because this is a methods paper, you know. And so the first thing he had to do is to take one strain of mice and immunize them with another strain. So he used C57 Black 6 mice as the recipients, and the immunizing strain was they were histoincompatible, so that they would, if they were immunized, they would reject the skin grafts and kidney grafts and whatever. And so he used a tumor cell that was called P815, and that was from a DBA2 mouse. And that's important because that's what Brunner had used. He'd used this P815 tumor cell in his colony assay, his cloning assay. So then they just waited 8, 9, 10 days and took the spleen cells from the immunized mice and teased apart the spleen and got them a single cell suspension, and then counted the cells and resuspended them at a pretty high density, 20 million cells per ml, 2 times 10 to the 7th cells per ml. In Eagle's minimum essential medium, you'll see a lot of that, with 10% fetal calcium. And then it took the target cells and labeled them with chromium 51 by a 30-minute incubation at body temperature, 37 degrees. Then he washed those cells 5 times by spinning them in the centrifuge and then resuspending them in fresh medium and then spinning them again and so forth. And then he mixed them together at 100 sensitized lymphocytes to 1 tumor cell ratio, 100 to 1, 2 times 10 to the 7th cells per ml versus 2 times 10 to the 5th cells per ml. And he showed then and then what he did was that you could stop the reaction at different time points. You could spin down the cells and take the supernatant off to see if there's any chromium released into the supernatant. And you could also then take the cells and do a colony assay to see, because that was sort of their standard at this stage of the game. So those were the methods. And the results were that they found when they used this ratio of lymphocytes to target cells of 100 to 1 and stopped the reaction after 3 hours, 6 hours, and 9 hours, that they got 50% chromium release after only 3 hours. This is good because that meant that those cells were being killed or something bad was happening to them quite rapidly. And if they used normal lymphocytes that had not been sensitized in vivo, they wouldn't get any reaction at all. So that was really important. So then they also compared it with they did a series of experiments comparing the chromium release assay with the cloning assay, and they got 90% lysis or death from the cells after only 6 hours. So that was also very, very good. And they thought they were on a sound footing. And then the other thing, they did a series of experiments where they varied the number of lymphocytes to target cells from 100 to 1 to 200 to 1 to 500 to 1. And as they diluted out the sensitized lymphocytes, they got less and less lysis, and that made sense. And so it was a quantitative relationship between the killer cells and the target cells. They had generated antibody against the alloantigens in the sensitizing cells. So they used these antibodies to see if it would participate in the killing reaction one way or the other. And they found out that the antibodies would block killing reaction, and it was antigen specific. If they used antisera to some other alloantigen, it wouldn't block them at all. And so that was good. It was an immunological kind of phenomenon. And they also fractionated the antibodies into the IgG fraction and the IgM fraction. They both worked. And then they thought, well, let's see if we can semi-purify antigens from the surface of the target cells and see what would happen if we use those instead. And when they used they made membrane preps really with lipoproteins extracted from the membranes, and it didn't have any effect whatsoever. So that, and they didn't know what they were doing anyway with regard to the membrane antigen business. We still don't. Here we are, 4 decades later. Then they wanted to. It had been shown by other people that in this phenomenon that other people had thought was a lymphocyte-mediated cytolysis, and the abbreviation for that is LMC. That you needed metabolic activity on the part of the killer cell. Yeah, and so they used drugs to block RNA transcription, and they also used drugs to block protein synthesis. And in both of those instances, when they used those compounds, they were able to block the reaction. And so that fit with the whole picture that there's something going on. These killer cells are sort of like, they've got their bayonets fixed and they're armed and ready to go, essentially. As to exactly what was going on is another story. But that's it in terms of this particular classic paper. And in the discussion of this paper, they said, well, they speculated. They speculated that the lytic effect, they thought it was lysis, actual lysis, and they thought it was damage to the membrane of the target cells. And somehow the membrane was made permeable to the macromolecules that were labeled with the chrome because they could get out. This wasn't just small molecules diffusing through the membrane. The membrane was not very intact, essentially. And they also had people had shown before that once the cell was labeled with the chromium 51, and if it leaked out, it was still attached. The chromium 51 now was changed. It was still attached to the macromolecules from the cell, and it could not be reutilized by the killer cells, for example. So that was also good in terms of this technique. And based on the numbers that they got and how they analyzed their data and their interpretation, it looked like it was a single hit phenomenon, meaning that a single lymphocyte could kill a single target cell. And that was also very interesting and important as time went on. They speculated that, they didn't know what was happening. They figured, they saw sort of under the microscope, they saw killer cells and target cells sort of together like they were kissing, but they didn't see that all the time. And they wondered what was happening. And they wondered, well, maybe there's a soluble molecule, some sort of a mediator molecule, like a lymphokine, that might be released by the killer cell that killed the target cell. That was one possibility. Another possibility was that the killer cell somehow, in touching and binding onto the target cell, stuck a hole in it with its bayonet. That's another option. And so they were pleased about their antibody data, and they were pleased about their metabolic inhibition data. And so that was good. And they went on to say then that the advantages of this technique were severalfold. One, it was simple technically to just do this. You mix together the cells, you spin them down, you take the soup. You don't have to be a PhD to do this, essentially. Even an MD could do it, right? So the other thing, though, it was very accurate, and it was very quantitative because you counted the counts per minute in the radioactive counter, like a Geiger counter kind of thing. So you got printouts then of the data of the counts per minute. So that was good. You could sit down and analyze them and so forth. And finally, this assay would take place just in a short term over just a few hours. And I think I mentioned in one of my other videos, when I've learned how to do this, maybe half a decade later, I liked this assay a lot because I would set the assay up in the morning. This is when I was at Dartmouth. And then I'd go in the wintertime, then I'd go off skiing. I drive to the Dartmouth skiway in like 45 minutes. I could ski up and down the mountain all afternoon, and I could come back and harvest my experiment and then go home and have dinner. That was good. I like this assay. So this became really, as I said, the workhorse of future studies. And they said this at the end of their report. This assay can be used for future studies of allograft immune reactivity and also tumor immune reactivity. And it did. And for the next 10, 15 years, even longer, this was the sine qua non of cell-mediated immune function, cell-mediated immune lysis of target cells. And therefore, it was subjected to all kinds of experiments using it, but also experiments trying to find out what exactly was going on. And just as for a little bit of a look ahead, so this is 1968 when they first published this. It wasn't until 1980 when the first, and it wasn't because people didn't try. I mean, people were trying to figure out what was going on. In 1980, a group at the NIH found that if they exposed target cells to immunized lymphocytes and waited a little while, and then processed the target cells and looked under the electron microscope, that they had big holes in the membrane, big enough to drive a truck through in molecular terms. And so that was interesting. And then just a few years later, another group got involved, and they did similar kinds of experiments with the electron microscope and could show that the holes were caused by tubes. Actual pipe was inserted into the membrane of the target cell. And also the immunized lymphocytes a few years later, and we're talking about the 80s now, and we saw this when we first generated cytotoxic T cells and got long-term T cell clones and so forth. When we looked at them under the microscope, they were chock full of granules, and which made them, they didn't even look like lymphocytes. It looked to me like they were macrophages. And I thought, I didn't think we really had cytotoxic T lymphocytes. Well, a group came along and they used our long-term T cell lines to grow a billion cells. And then they broke those cells open and purified those granules. And then a few years later, they analyzed the proteins inside those granules, and for the first time identified a protein that subsequently was called perforin, which formed those big pipes that got stuck in the membrane. And so those are all reports that are yet to come, but it gives you some sort of an inkling about how important this first paper was back from 1968. So I'll stop there, and don't forget, go on to the YouTube channel and like this video and subscribe and get notified by ringing the bell, and check out everything else on the website. So thank you very much.